Hello everyone, in this video I'm going to talk about the Lac Operon, and I will also explain to you how you should write your lab report over the Lac Operon lab that we completed. So first of all, what are operons? So in prokaryotic organisms, genes that are related to a particular function are usually clustered close to each other. And this is very convenient because if the genes need to be transcribed, they sort of work as a switch. They all get turned on all at once, and everything that needs to be transcribed and translated can be done pretty much all at once. So this serves as a switch of on and off for biochemical pathways within prokaryotic organisms. Now, an important operon in E. coli is the Lac Operon. This is the portion of DNA that enables the bacteria to break down lactose for energy. Now, glucose is the most preferred source of energy for E. coli. And if glucose is available, this system over here is going to be turned off. However, if lactose is available and glucose is unavailable, the Lac Operon makes it possible for E. coli to metabolize lactose for energy. So these are some portions of the Lac Operon that you should be very familiar with, not only to write this lab report, but also for your exam. Make sure that you know what the cap site is, what the cap molecule is, what the promoter is, what the operator is, what a repressor is, and you should also know the Lac Z, Lac Y, Lac A, and what they code for. So all of these are important to understand. So first, let's start by talking about the structural genes of the Lac Operon. These are the structural genes of the Lac Operon. We have the Lac Z, Lac Y, and Lac A. You should know what they code for and what the functions of those proteins are going to be. Again, these are called structural genes because this is the portion that gets transcribed into mRNA, which then gets translated into some sort of functional protein. So first, the Lac Z encodes for an enzyme called beta-galactosidase. Beta-galactosidase can cleave lactose into galactose and glucose. As you can see here, lactose is a disaccharide. So it is made out of two sugar molecules that are linked together. And in order to metabolize lactose, the cell needs to break it down into galactose and glucose, and it uses beta-galactosidase to do that. However, beta-galactosidase can also convert lactose into allolactose and allolactose back to lactose. These two molecules are isomers, which basically means that you could think about them as the same molecules that are basically arranged differently in space. Lac Y is going to encode for Lac permease. This is the permease over here. And as you can see, what Lac permease does is that it allows E. coli to pick up lactose from their environment and bring it into the cell. So here's lactose, it is going to move into the cell through the permease, and then if beta-galactosidase is available, this lactose can be broken down into glucose and galactose. Or beta-galactosidase can convert lactose into allolactose. And then there's also Lac A, which is going to encode for transacetylase, which is also another enzyme that is important for breaking down lactose. It helps convert galactose to glucose. So those were the structural genes. Now there are parts that are really not used for like translation and transcription, but they are important parts of this DNA molecule and important part of the operon. So there is a region called the promoter, and this is a sequence of DNA which the RNA polymerase binds to. So it attaches to it right here so that it can then slide down this DNA and transcribe this portion of the DNA. So that's what the purpose of the promoter region of the DNA is. It is a binding site for RNA polymerase. There's also something called an operator in this operon. Here are as you can see, these over here are going to be the structural genes, and this is the promoter, which is the binding site for RNA polymerase. In between the structural genes and the promoter, you have this portion of DNA called the operator. Now, the operator is sort of like a switch. If we don't want RNA polymerase to transcribe, we can place something on the operator here to block it. If we want it to transcribe, we could remove the block and allow it to start this process of transcription. So this molecule right here, by the way, this is a repressor because it binds to the operator and it prevents the RNA polymerase from transcribing. In conditions where there's plenty of glucose for the E. coli to metabolize, the repressor is going to be bound to the operator. However, if lactose is available in the environment, it enters the cell and it will get converted to allolactose. Allolactose is going to bind to the repressor. As you can see, the repressor right here, it has two binding sites. One is over here and one is over here. So it will bind to the repressor, it will cause it to change shape, and it will fall off the operator. So up here is a condition where glucose is present and lactose is absent. When glucose is present, the repressor is going to stay bound, and there's no lactose, so there's nothing there to remove the repressor. So the repressor is bound, and there is going to be no transcription of this operon over here. However, if glucose is present and lactose is present, lactose is going to enter the cell. It's going to bind to this repressor, remove it. But if glucose is still present, there's not going to be much transcription of this Lac DNA region, this Lac Operon. And that is because of a molecule called cyclic AMP, which we will talk about in just a second. However, if glucose is absent and lactose is present, then this gene is going to get transcribed in very large amounts. Okay, so again, if glucose is present and lactose is present, the repressor here is going to be removed by allolactose because you're going to have lactose entering into the cell. Lactose will get converted to allolactose. It will come up over here, bind to the repressor, remove it from the operator, and free up this region for transcription. However, we need more than that for transcription to happen. We also need something called cyclic AMP. So what will happen is that if there is high levels of glucose and lactose, then there's going to be low cyclic AMP. So when there's a lot of glucose available, cyclic AMP is low. When glucose levels drop, when the cell gets starved a little bit, cyclic AMP becomes high. And cyclic AMP binds to this molecule called CAP, which is going to basically give the green light to RNA polymerase. So what we see here is that CAP right here needs to bind to cyclic AMP, and cyclic AMP is going to move here, and it is going to give RNA polymerase the green light to start the process of transcription. So, as you can see here, if both lactose and glucose are available, lactose will enter the cell and remove the repressor, so the operator region will free up. However, cyclic AMP levels are going to be low because glucose is still available. So there's not going to be much transcription happening here. However, if we have only lactose available, lactose will enter the cell, it will remove the repressor from the operator, so it will free up this portion of DNA. And also, because there's no glucose available, you're going to have high levels of this particular type of molecule called cyclic AMP. When you have a lot of cyclic AMP, it will come together here with CAP molecule, and they will come over here in this promoter region and give RNA polymerase the green light, and there's going to be a lot of transcription. So the way you could think about it is that there's two things that are happening here. There can be a roadblock here on the operator, and you also need the green light. So for transcription of the this LAC portion to happen, we have to first remove the operator, I mean the roadblock, which is the repressor, and then we also need the green light. So lactose entering into the cell will remove the block from the operator and free up this region for transcription. But starvation from the absence of glucose will increase the levels of cyclic AMP, which then binds to CAP, and together they're going to come here and give RNA polymerase the green light. And then high levels of transcription can happen. So we need both of these conditions for a large amount of transcription to happen. I do want to mention that in this lab we will be also using a molecule called IPTG, which is a mimic of allolactose. So it kind of like looks like lactose. It will bind the same way lactose does to the repressor. The difference is that it is a superinducer, which means that when it binds to the repressor, it will bind irreversibly. It won't come off of it. So when we have IPTG, you're going to have large amounts of transcription of this Lac Operon region. However, IPTG, even though it binds to the repressor, it cannot be metabolized. So IPTG will not get metabolized, but lactose, on the other hand, while it removes the repressor from the operator portion, it can also be, that allolactose that is bound can also be metabolized for energy. And I do want to mention that lactose is considered the inducer for the Lac Operon, because you have to have lactose to have it enter the cell and then remove the repressor from the operator portion. And we did talk about CAP. CAP is a positive transcription factor or activator, and CAP and the coactivator cyclic AMP need to come together so that RNA polymerase can receive its green light for transcription. In the lab that we did, we looked at cell growth of E. coli in 4 different conditions. We had a culture of cell and we put them in 4 different conditions. The first one was in glycerol, the second one was in lactose, the third one was in glucose, and the fourth one was in IPTG. The results that were received were not ideal. However, we will work with what we have. The procedures are going to be written in your packet, so you could use that for the method or procedure section of your lab report. And remember, depending on your group, you either had the wild type bacteria or you had the mutant. And you guys were supposed to exchange data by the end of the class. So if you had the mutant, make sure you ask for data from another group that had the wild type, so you have both data. If you had the wild type, ask another group that had the mutant, and you gotta have both of them. In protocol 1, we were pretty much looking at in what types of conditions do bacteria grow and how fast they grow. If it was a wild type, as you would guess, probably the glucose would be metabolized the fastest, and next lactose, and glycerol wouldn't be metabolized at all. And the mutant, it really depends what the mutation was. Initially in this lab, they had a condition where it was lactose and glucose, but it was taken off for our lab, so you don't have to worry about including this. Your graph should look something like this, but again, if it's not like this, because the results weren't ideal, that's perfectly okay. So what you're gonna have is 4 different graphs in your lab report. For protocol 1, please go ahead and have a table for the data that you collected in lab. This is from your first, you have to include data from your own lab. So what you collected from your group and the other group where you're going to be exchanging data from. This is probably incomplete. From what I remember, we only got maybe this far or this far. That's fine. Go ahead and put that table into your lab report. And for this missing stuff, just put blank. And I want you to write in your lab report that we did not have sufficient amount of time to complete the lab. So therefore, the data that we received are incomplete. However, this is data given to us from the Monday lab that we're going to be using for graphing. So for cell density, remember cell density, we measured their growth over time and for different conditions. And this is going to be wild type. One table is going to be for wild type. Another one is going to be for the mutant. So this is the one for the wild type. You're going to also go ahead and enter in this table into your lab report. And what I want you to do is to graph this on Excel and then paste it into your lab report. The x-axis will represent time, the y-axis is the cell density. And if you're not sure how to use Excel for graphing, go ahead and just look it up on YouTube. It's actually quite easy. On your graph, you should have 4 different lines. And make sure you either color code or label the lines so we know what they are. And same thing here. This is also cell density, but for the mutant bacteria, this is the results that I got from one of the Monday labs. And you're also going to make a table for the mutant. So again, you should have 4 different lines. And whenever you want to find out what the mutation was, what specifically was mutated, you could go back between these two and kind of compare the results and come up with a conclusion, like a logical conclusion over what you think might have been mutated in this bacteria. So that was cell density, has to do with protocol 1. And for protocol 2, we were going to look at the enzyme activity of beta-galactosidase. And we do that with this chemical here called the ONPG. And the ONPG, it's sort of this chemical here that whenever it gets broken down by beta-galactosidase, it will give you ONP and galactose. So what happened here is ONPG was added to all these different conditions, and then under the 420 absorbance, we kind of looked at what the absorbances were. So what basically happens here is that when we add ONPG in these conditions, depending on how much beta-galactosidase is available, you're going to have a certain amount of ONPG. If your cell, if under those conditions there was a lot of beta-galactosidase, then you're going to have high levels of ONPG. If beta-galactosidase was low, there wasn't much of it transcribed and translated, ONPG would be low. So high ONPG, high beta-galactosidase. Low ONPG, low beta-galactosidase. So here is the results for the wild type and under these 4 different conditions over here. And here it is for the mutant. And you could kind of compare these two, wild type and mutant, to determine what the mutation could have been. So I'll give you like an example of one over here. As you can see here, the wild type in the IPTG condition, the beta-galactosidase activity was actually quite high, as you see over here. But when we go over here to the mutant, there was barely any beta-galactosidase activity right here under the IPTG. So this tells me that for whatever reason, the Lac Z was not being transcribed or translated. So possibly a mutation in that portion, but that's something that you're going to have to decide. And here is the ONP standard curve. We didn't actually do this in class. It was done for you. This is these results are achieved by serial dilution. So you could just put in this chart for protocol 3, and here is the ONP standard curve. You could go ahead and actually copy and paste this in your lab report. But then again, I just want to reiterate that right here for protocol 2, here's ONP over here. And the more ONP we have, that indicates higher beta-galactosidase. So the absorbance, whenever we measured the absorbance of these, the higher the absorbance, the more ONP was available, which means that there was more beta-galactosidase. Okay, and for your conclusion, do the best that you can to determine what mutation was involved and give a logical explanation for that.